Hello, everybody, and welcome to the Emerging Technologies Information Sessions 2025 webinar series. This is our final week of presentations. My name is Sam Cimino. I work for the U.S. Geological Survey and the Pacific Northwest Aquatic Monitoring Partnership. And over the last year, a planning committee has been putting together this ETIS webinar series, but we didn't start with a blank canvas. The Pacific Northwest Aquatic Monitoring Partnership and the Pacific States Marine Fisheries Commission's StreamNet program have been hosting various versions of these emerging technologies conferences and webinars for more than 14 years now. And the goal of these emerging tech projects is to bring together monitoring professionals, project managers, and data managers to showcase how they implement the latest technologies or old technologies in new ways. And like I mentioned before, we are in our final week of this 5-week webinar series. And today's session will cover 3 presentations on the latest trends and tools in remote sensing. And these sessions are all being recorded, as you probably saw when you joined. So if you missed out on any of the first 4 sessions, you can check out those recordings on the PNAMP YouTube channel, and these will typically be available within the week of each session. Actually, hopefully a little bit sooner than that, within a day or 2. And now I'm going to kick it over to our moderator today, Meg Detloff.
Hey, good morning, everybody. Like Sam said, I'm Meg Detloff. I work with him at USGS and PNAMP. Thanks for joining us for the last session of the ETIS webinar series. I'm going to cover some housekeeping stuff. Sam, do you have the housekeeping slides? There we go. On your screen, you should see some of the commonly used icons and teams. Unless you are speaking, please turn off your camera and microphone. If you have questions or feedback, please use the chat function or raise your hand and unmute yourself. If you don't have access to chat, which happens sometimes with some entities, please feel free to unmute or raise your hand. We can mute you, but we can't unmute you. Like Sam said, we're recording the meeting and it'll be available on our YouTube channel this week. A couple additional troubleshooting tips. If you're having trouble with functionality, try leaving and joining again. Use a different browser. Edge usually works better for Teams than Chrome does. Or try using the desktop version instead of the browser, and vice versa. Slide. PNAMP commits to fostering an open and welcoming environment, so please remain respectful. Righty, Sam. Okay, so let's begin. Let's see, Ed, are you out there?
I am.
Perfect. All right. So just a note before I hand this over to Ed. After each presentation, we'll have about 10 minutes allotted for questions, depending on time. And if we have extra time at the end of all the presentations, we'll open the floor again for questions on any of the presentations. And so while Ed is getting set up, I am going to give some background on him. All right. Dr. Rudberg is a seasoned CEO who has brought multiple companies from product ideation to profitability. Through this work, he and his companies have been identified as a thought leader in water resource management. He has a PhD in natural resource science and management and a master's in marine affairs and policy. And with that, I will hand this over to you.
Great. And just to double check that, is everyone seeing my presentation now?
Yep, looks great.
Great, thanks so much. So just a bit of a background. We are developing a biosensor that's based on Droplet digital PCR, and I'll go through a little bit of what that means. But it's essentially the world's first continuous autonomous sampling Droplet digital PCR machine. And so, I'm not telling you all things that you haven't heard of before, but we as a society are facing environmental and health catastrophes without the real-time detection of biological species of concern. And for what we're talking about today, what's probably most important is in our natural resources and oceans, where invasive species cost the world's economy in excess of $400 billion per year. That's both terrestrial and aquatic. We're obviously focused on marine and aquatic. And then also with aquaculture, that might have a lot of overlap with the work that you all do, where disease costs host producers in excess of $5 billion per year. And in water and wastewater, the inefficient management of wastewater results in a 100% increase in greenhouse gas emissions and great efficiencies that if we can make our wastewater treatment more efficient, our infrastructure can last longer and have less concerns with for human health. And something you all aren't probably interested in, but on biomanufacturing, as we look to the future for foods, fuels, et cetera, there's just a large inefficiency that comes from an inability to monitor what's happening in vessels. So what we envision is a world in which we have, particularly in the ocean and water environments, along with biomanufacturing, an array of sensors that not only are looking and quantifying biology in the fisheries, but also in our marine protected areas, in our nearshore environments and wastewater, in areas that are having offshore wind development, so that we can monitor the success of mitigation, the success of conservation, and really trying to manage the spread of species of concern most effectively. So just to give everyone a background, I'm sure the vast majority of people under here know what DNA is and know what PCR is. So a little bit of unnecessary background, but to make sure we're all on the same page. PCR is used to amplify genetic material and amplify DNA. So 1 becomes 2, 2 becomes 4, et cetera, to the point of where one can detect the presence of DNA of concern. But what we're looking at is a specific type of DNA in the environment. We break down DNA, or environmental DNA, and RNA into 3 types of environmental DNA. First is cell-associated. Fish swim around, they do their little onesies and twosies, they have slime, they're shedding cells. We're all shedding cells into the environment. And that is what's actually typically looked at when we think about eDNA monitoring. Same thing with particle associated, but particle associated, the cell opens up and lyses, and the DNA can attach, or cells can attach to particulates in the environment. So if you think of when you draw a sample through a filter and you test that filtrate, that is the type of DNA that you're looking for. We're looking for something very different. We call it free environmental DNA, in which the cell lyses and the DNA is spilled out into the environment. The issue with detecting free environmental DNA, or RNA, is that it's a very short-lived biomolecule. Phosphorus is food for many things in the environment, and therefore it just doesn't stay around very long. Which is good and bad. The good thing is that you know that it's something that was likely nearby recently. But the bad thing is that it needs to be analyzed in the field because of its short half-life. Just to give everyone a little bit of an introduction to Droplet digital PCR, as compared to a typical PCR where you're, think about, hey, we swabbed our nose during COVID, right? We get a 1 or a 0. It's, yeah, you have COVID, or yeah, you don't. As compared to what ddPCR can show us is how much COVID someone has. So what it amounts to is having many different discrete PCR reactions that either become positives or negatives, 1s or 0s, that thereby we can quantify DNA at the end in concentrations which is gene count per microliter. So if you think about typical sample collection, when we talk about cell-associated or particular particle-associated environmental DNA, you have a water sample, you filter it, you lysate it, you separate it, and you go through your PCR to get the environmental DNA. For our technology, we've taken out that lysate and separate step, because we are allowing free environmental DNA to pass through our filter, and thereby we are, as I said, detecting that free environmental DNA. Well, we're using a technology that is emulsion microfluidics. So what would happen is that water that would come into our sample, we mix it with our primers and probes, and then we make those discrete samples with an oil and the water. So it essentially, in a microfluidic tube that is smaller than the width of your hair follicle or your hair, you get very small discrete bubbles or droplets that we then have in that go through PCR and get analyzed for their glowing. So as compared to a well plate of a droplet digital PCR machine, that each one of those wells will have the 1 or 0 in it, we have 1s and 0s in these discrete droplets. And so what we have to date is a alpha prototype. To date, we've been supported not only with the USDA SBIR1, but a NOAA SBIR1, 2, and now we have a current project for a NOAA SBIR3 that has been developed, the technology. It came out of the University of Montana. And really our goal for our technology is to have continuous sampling every 15 minutes, as often as every 15 minutes, with end-to-end autonomous operation. That means we've taken the human out of the equation and we've put a laboratory into a box and have that box being able to be taken to the field where it can quantify DNA or RNA concentrations. And we can also therefore give geotemporal data that can show trends over time. So if I'm over here and I swab my nose 20,000 times versus I'm over here and I swab my nose 20,000 times. And currently we can detect up to 3 species at the same time, with our goal as we go to commercialization up to 5 species at the same time. And as we think about the development of our technologies, we are really focused on those key metrics for UN sustainability, and you can see those below, whether that's water and sanitation or life below the water. So how does it work? Well, you know, one of my pet peeves for presentations is for folks to show a very complicated slide, which of course, therefore I'm doing and apologizing just like everyone else does. But I'll give you the really high level. We talked about I talked about a little bit before. The idea is you take that water sample, you mix it with those primers and probes and everything that one needs for that reaction formation. Then we make those from 20,000 or 20 microliters, we make 20,000 subsamples. So 20,000 discrete droplets that then go through the thermocycling process, and either they glow in one of 3 colors or all 3, or they don't, which allows us then at the end to quantify the data and push it up to the cloud so that you can get genetic concentration in your phone, on your tablet, on a desktop. And the questions often come up as well, what about decontamination or the risk for cross-contamination? Well, we have the ability to, if you have a positive that you're concerned about, go through and decontaminate the system and then run a sample again. The reason we wait for every 15 minutes is you think about these droplets like airplanes on a runway. We have to have a separation between those airplanes so that we can identify sample 1 versus sample 2. Question always comes up around accuracy. We always say we stand on the shoulders of giants. While we can have as little as 1 molecule of DNA detected in a sample, it really comes down to the primers and probes, how they're designed, and making sure that they've been vetted in academia and governmental agencies or others, because we're only as good as the primers that we put into our machine, because we're entirely species agnostic. So you could bring out, quagga zebra mussel, you could put in, you name it, invasive species, bloody red shrimp, or invasive carp in the upper Midwest. So really our goal is to provide real-time data that delivers key benefits to our customers, and that's early detection. Between sample and answer of less than an hour, and ongoing sampling without effort. And the goal is to have more data that results in better management and rapid response, and thereby can safeguard human health and the environment while saving on operational costs. How do we differentiate ourselves from other technologies? Well, typical technology is you take your water bottle, you take a sample, you send it off to a laboratory, wherever that is, and get results in weeks, if not a month. We can give sample to answer within the hour. And the idea is that those real-time results can power things like AI, machine learning, and give really detection reliability. We all are, I hope, familiar with type 1 and type 2 errors. The possibility of detecting something in error that's not there or missing something that is there. And by using ddPCR technology and the number of samples we can take, we hope to really reduce that risk statistically nearly down to zero. And I would say ease of use, right? For those of us that are of my generation, maybe you remember Ronco and his food dehydrator or rotisserie, and the idea is set it and forget it. And so that's really our goal, is the ability to put something in the field, have it set up, and then it samples at a predetermined amount of time. So look back at where we've been and where we're going. The version 1 of the technology, like I said, came out of the Flathead Lake Biological Station. The original inventor of the technology is Dr. Cody Youngbull. And you'll see a picture of that there on the shore of the Flathead Lake. And with that original technology, it had to be fed samples that were already, had the primers and probes, everything mixed up, and then it would spit out an answer for you, or results rather. And this is an example of our environmental water sample, looking at quagga zebra mussels versus a no template control. And this is one way we can show the data, where you have fluorescence on the y-axis and time on the x-axis. So each one of those little spikes is one of those droplets that's going by the detector head. And when you have a positive, you'll see that it fluoresces more and it glows. And when it has a negative, it goes below. And you'll see there that there is a green line that we can either set automatically or manually so that we can detect and differentiate the positives from the negatives, the ones and the zeros. And so you'll see that on the environmental water sample, they had 69 copies per microliter versus a no template control, so deionized water, you get 0 copies. All right, technology, it's also very important to have lab tests to show that you're expecting results from what you're getting. So we put together a known level of concentration, taking a look at what we detected, and we got linear results. So it really makes us feel like we can really rely upon the results that we're getting. Here's another case study that was done in partnership with the National Park Service, who provided boats and support for the project, looking at Lake Mead and going from Site 1 to Site 7, as to be expected. At Site 1, high concentration, over 9 copies per microliter. As you go to Site 5, you'll see the decrease down to just above 3 copies per microliter. And then as you come back to Site 7 in a marina, you're seeing the high concentration again. A marina, you have pilings, many different substrate that quagga and zebra mussels can live on. Same type of experiment done in Lake Travis, Texas, starting at the marina, seeing a very high concentration of quagga and zebra mussels. Going out towards the center of the lake, you see it decrease. And then we saw it increase again, and we were kind of surprised by that, and realized, though, that there is a buoy out there, again, providing substrate by which quagga and zebra mussels can live on. And at the same time, looking at Lake Mead, looking at pre and post decontamination. Again, you're seeing the fluorescence on the y-axis, and with these samples, they were taken directly from recreational vessels, recreational boats, and you're seeing some depression there in the fluorescence. And something we call inhibitions. Things like gasoline are an inhibitor of droplet digital PCR technologies. However, because we can set the threshold between positives and negatives, digital droplet PCR technology is rather very robust against inhibition. And so you're seeing before and after decontamination. This is another way to display the data. We are seeing that we have counts on the y-axis and then peak height. So that's how much did it glow, right? Fluorescence on the x-axis, count on the y. And this is supported by our NOAA SBIR2 work in conjunction with Pacific Seafood, looking at columnaris disease and proliferative kidney disease in aquaculture. And what we found is that you get a bimodal distribution of the data. So there's your ones on the right, your zeros on the left, and then we can set a threshold in between. While they're interested in columnaris disease, it's important to note that they are interested in more than the absence or presence of the disease. They are interested in changes in concentration over time because it's something that's omnipresent. Here are the results from a USDA SBIR1. We're looking at incorporating reverse transcription, so the ability to detect viruses and RNA. And what we found is that using what's called a one-pot reverse transcription, we can preheat the samples and then feed them into our machine so that we can use what's called cDNA, so copies of DNA again, showing amplified droplets on the right and unamplified droplets on the left. And so as we move forward in the future, our goal is to integrate reverse transcription because we now have proof of concept of the ability to do so. So our current work, which, to be frank, is supported by the Inflation Reduction Act. So we're not entirely certain how that it's going to impact our project. It changes a little bit every day. But the overarching goal of the project is to look at Batavia Bay in southern Louisiana and looking at bottlenose dolphin post some of the work that have been done there for environmental restoration. We're also partnering not only with NOAA, but the US Navy for another site in San Diego, looking at a site that would more or less be a control where they're already training dolphins so that we can feel very confident. We're also looking at human waste indicators. So the ability to look directly at things like human E. coli, et cetera, in the coastal areas, but also red snapper. Some of that may change based on the impacts of funding, but the project as we know it is still moving forward. So just to give you a little synopsis on where we're at versus where we're going. We started at the University of Montana. We had sample preparation, which was external. Again, the machine had to be fed samples. We could do one sample every 15 minutes and look for one species at a time. As we move forward, though, our current alpha prototype that was funded by NOAA for the SBIR 1, 2, and now 3 program is 3 channels. Everything for sample preparation is happening internally. And unfortunately, our power source is still external. But as we move forward to commercialization and our beta prototypes, we're going to increase our species detection up to 5 channels, include reverse transcription for RNA detection, as well as in the future, aerosol detection, because we have IP intellectual property around the ability to detect aerosols. And then take it off grid with battery and solar for more remote locations. So as we transition the technology, our overarching goals are to expand pilots and love to talk with those that have interest in doing pilots in the future. That's including drinking water, wastewater, et cetera. Look to commercial production and RNA quantification with reverse transcription. More deployments, faster reaction speed. Overarching as we continue this technology. We call it our big ugly baby right now. And so our goal is to have something that is smaller, faster, and more robust, and participate as much as we can in fundamental eDNA research. Many questions remain about correlations between environmental DNA abundance and organism abundance. And there are just a lot of fundamental questions we would like our technology to help be able to solve. So with that, I wanted to thank our founders, the Flathead Lake Biological Station, NOAA, Forest Service, U.S. Fish and Wildlife Service, USDA. The technology came out of an NSF-supported laboratory at the University of Montana. And with that, I will open it up to questions. And it looks like I didn't go over time.
No, you did great. Thanks, Ed. That was a really cool presentation. You guys are doing some amazing work. And I hope to never have to swab my nose 20,000 times for COVID.
Right?
So if you guys have any questions for Ed, feel free to type them in the chat. I can read them, or raise your hand and we can call on you, or you can just go ahead and unmute yourselves. Quiet crowd this morning. Go ahead, Sam.
Yeah, hey, Ed. Great presentation. You kind of mentioned this, and I think you also mentioned, you're using solar power to help keep these out in the field longer, but how long can they stay out in the field without being recharged or, some sort of like maintenance visit?
Yeah, so our goal long term is over 30 days in the field. So right now we're plugged into the wall. So we still have that development to go on the battery and the solar, but from a primers and probes perspective, our goal is at least 30 days in the field.
Very cool.
Thank you.
Other questions?
All right, we have a question from Doreen McCoy. What is the cost of a detector, and also what training is involved to use them, and what kind of ground truthing sample? What are the ground truthing samples looking like?
Yeah, so the first question is cost. We're not commercialized yet. I'll be frank. So we're very much still in our pilot mode. Our goal is to, at least in the beginning, be on par with other droplet digital PCR technologies. We hope to reduce that cost over time. The other is how do we ground truth. We use things like G-Block and others to ensure that we can have within our system controls, whether that's a negative control in the form of deionized water or positive controls. So that is one thing that we can ensure the technology is robust and giving you data that you would like. And I'm sorry, there was a second part of the question that was right in the middle that I didn't answer.
What kind of training is involved to use them?
So we're trying to make this as simple as possible as we move forward. The primers and probes are really what is going to be the thing we have to make as easy as possible. So our goal is to make them like printer cartridges so that someone can put it in the field, turn it on. Where the training really is going to come is the person that's analyzing the data on the back end, right? Because we can give you counts per microliter, but depending on your distinct goals and what the type of work you're doing and what you're looking for, the management repercussions are a lot going to be on the user and their goals.
All right. Question from Denise Kelsey. How big and how heavy are the detectors?
All right. Well, right now, like I said, it's our big ugly baby. It's suitcase size. So under 45 pounds, but big and unruly. Our goal as we commercialize is to have it be the size of a carry-on. It's got to be mobile. It's got to be able to be picked up by an individual and moved around, regardless of all of our physical abilities.
All right. Sounds like quite an effort to try to get that compacted down.
Actually, one of the easier parts. We just built these ones big so that we can get our hands in there and reach, and do things. So getting it smaller is actually one of my least concerns. So I'm excited to have something small that I can put in carry-on and see what TSA says.
Yeah, probably wouldn't be too fond of that, but good luck with that.
We've done it before. We just have to call ahead and say, hey, we're bringing, and it's like, okay, they're expecting us, and they go, they go through and swab and do all the things that they need to do. But what do you do?
That's pretty cool. Yeah, I didn't know you could call ahead and warn them. All right. Anyone else? You can unmute yourselves. All right. Feels like it's winding down.
Okay.
Ed, are you gonna stick around for?
I'm gonna stick around. I'm also going to put my email in the chat so that if questions come up later.
Okay, excellent. That's great.
Thanks, everyone.
Thank you so
Appreciate
much.
your time.
Yeah, that was an awesome presentation. Thank you.
Thank you.
All right. Next up, we have Courtney Zambory. And as with Ed, I'll give you background on Courtney while she's setting up and then hand it over to her for her presentation. Courtney Zambory is a spatial information analyst for the Oregon Department of Fish and Wildlife's Fish Research, Evaluation, Data, and Decision Support Group in Corvallis. She received her master's degree from Iowa State University, where she focused on developing novel geospatial methods to support aquatic conservation. Since joining ODFW, she has continued to creatively leverage spatial and other analytical techniques to advance and support Oregon's management of its diverse natural resources. And with that, it's all yours, Courtney.
Thank you so much. Hi, everyone. My name is Courtney Zambory, and I'll be talking today about a GIS tool that I created as part of a contract work with ODOT a few years ago called the Early Prediction Method for Native Migratory Fish Presence at Small Culverts, which is a mouthful. Let's see. But this work started as an effort to provide a decision support tool that would help biologists out in the field to do an initial scoping and prioritization of where they should start looking for fish and whether or not these culverts across the landscape would trigger a fish passage law. That law means that if the culvert or a barrier is thought to be a barrier to one of the 32 native migratory fish, then it would require some additional work to make sure that it met the passage requirements for those fishes. So this was a tool, again, that was created for ArcGIS. It's a plugin that works with ArcPro, and it's Python-based, so easily modified or tweaked if new data sources come in and scalable as new versions come online. So the Oregon highway system is what we focused testing this tool on. So 51% of the Oregon highway system has been surveyed for these culverts. This specific project focused on culverts that were less than 3 feet in diameter, and the initial data delivery from ODOT was over 12,000 culverts. So there was a need to start looking at these and seeing which ones were likely to trigger a fish passage law, which ones were not likely, and highlight areas that really needed attention and a field biologist to go out on the field and confirm for ODOT. We decided to stratify this work by these Huck 6s. So we worked on the coast and the Willamette Basin as our first 2 testing sites for this tool. So first off, we talked and surveyed with biologists that went out to go survey these culverts and kind of asked them to develop a flow chart so we could translate this to a computer, figuring out why or when a culvert would trigger this fish passage law. And so this is what they came up with. So the first section, and we'll go through this as we walk through the presentation, is there a defined stream channel upstream of the culvert? And then as we go through this, we looked at the flow chart, translated it into a Python script. So if or then statements, put it into ArcPro, and now you have a GUI that you can put in your own data, and it will pop out with whether or not that culvert was likely to trigger this fish passage law. So when you open up Arc Pro, you don't see any of the code that is associated with this tool, but rather this GUI. It says the Culvert Analytical Tool for the Willamette. There's also one for those different regions. And you put in a culvert point, so that's a point file. You can request that from ODOT. It should all have a unique identifier with them, and that tells the code to process each culvert individually and to export that decision based off the individual culvert. For this, our unique ID was called a DFID, and that was consistent with ODOT's naming conventions. A highway GIS line network, that really could be any line network that you choose that corresponds with your barrier. And it helps to have that because it creates the delineating factor to look at where the culvert starts and ends on either section of the road. A LiDAR digital elevation model, those are publicly available from the Dogami website. A input of a species distribution model, which we'll talk about again later on the presentation. A standing water polygon, which for this we use the National Wetland Inventory. The network that we're looking at for the stream network itself is based off the NHD high res. We converted that to a trace network. Instructions on how to do that are incorporated with the manual to this tool, and then finally a location of where those data, the final data set will be written. Okay, so we'll talk about the culvert or the data inputs as we go through the culvert data. This could actually be any barrier, again, that has that attribute, a DFID field with unique numbers in there. But for this, we look at the ODOT data delivery. And so we just asked ODOT, can I have a point file with this DFID attribute and then latitude and longitude across the landscape? So here's an example on the right of all of the culverts that we're looking at scoping for this project. So the highway network, publicly available. We just downloaded a very standard highway system from Oregon's Data Explorer. Doesn't have to have any special attributes to it, so nothing really special about it. It could be dissolved itself. Inputs as well. This is the LiDAR, so the digital elevation model. This tool needs elevation data around each of the culverts, but it does not have to be continuous, so you can mosaic it together, but it can be patchy across the landscape as long as it covers all of your culvert locations or barrier locations. This data set is not available for the entire state of Oregon, though it is being updated. I think there's more holes have been filled in since this presentation, so just check back regularly as you go through. This is what DEM looks like. So again, these little dots in the corners, those are the culverts, and I just downloaded and mosaicked the different tiles that covered those culvert locations. For a species distribution network, these are the 32 species that are covered by Oregon law that are considered native migratory fishes that would trigger a fish passage law. For us, we created a species distribution model across the network. For many of these species where data were available, where they were not available, we simply subbed in Oregon's native fish habitat distribution layer, which is on our ODFW website. So for the species that we did have enough data, we took all the sampling locations across all years. The fish passage law is triggered for historical habitat of species, even if species no longer exist there. So we took the entire record, presence and absence, and converted those. We associated them with the NHD high res. So on the left here, those presence and absences are in the black and white dots respectively, and then we translated those to the reach codes that were associated with them. So now instead of we have points, we have lines. And in this data set, there was a really even split. Ironically, for lamprey, that was about 50-50 presence and absences, which was really nice. We use a variety of covariates that were associated with the NHD high res. Anyone on this call who has used it before knows that one of the limitations is that it's not spatially or not density cohesive throughout its entire range. So this is an example of the Nehalem, where work has been done in the upper reaches, whereas less work's been done in the lower regions. So we chose for this modeling density independent covariates, which include slope, drainage area, minimum and maximum elevation of each one of those lines or reaches. Terminal path, essentially that just is a code that looks at the all the lines that have a common drainage point. Hydraulic class, path length, and power. Path length essentially is the distance from the terminus of the network, so like distance to the ocean essentially for these examples. Covariates also included climate data, so looking at mean fall precip, mean spring precip, the 30-year normal average air temperature, winter and summer precipitation, as well as, the winter and the summer air temperature. And then we also looked at upstream cumulative stream kilometers. So these are all covariates, again, that were density independent of the network itself. And then for our species distribution model, we used our package called BioMod, which is an ensemble modeling package. We selected 4 different model types and conducted a 75-25 split. So the data were trained on 75% of the model or the data, and then tested on the remaining 25%. And those, so we had 4 different models. We split them between that 75-25, and then we ran those models each 25 times for a total of 100 models, individual models run. And then we did a weighted average on the results of those models to get the final distribution probabilities, which range from 0 to 1. So this is what it looks like for lamprey results and coho results. So essentially you run the model across each one of the reaches, and then it gives you those scaled 0, which is a low probability of presence, to 1, which is a high probability of presence across the network. For standing water, which is an important input, and we'll talk again how these all coalesce together, is the National Wetlands Inventory. So we just use those downloaded straight from their website. No additional work has to be done for that input. There is a little bit of work that needs to be done beforehand, which is looking at the barrier database and looking at barriers that were blocked. So natural barriers that were fully blocked, and putting those into the tool itself. We define natural barriers for at least our barrier database, which does have some holes, but is the most complete barrier database for Oregon that's available, was cascade gradient velocity and falls. So those are naturally occurring. Again, we didn't consider artificial barriers because the fish passage law says that if a fish species was there or used to be present, then it would still trigger a law despite its lack of presence currently. So we put all of the the natural blocked barriers again on the network, and so those are highlighted in red here on the map of the Willamette. So essentially this is the stream network. We run it, we convert it from hydrography to a trace network with all those barriers on the network. And so now we can run the tool. So this is our, again, our support flow chart here that looks at if there's a defined channel upstream of the culvert, yes or no. So the way we define this is that we're going to look at the network, but our first problem is that not all the barriers, the culverts, land on the National Hydrography data set. So to comply with this flow chart, we have to figure out where water is flowing up and down across the landscape outside of just that standard hydrography. So this is where the LiDAR comes in. So we extract LiDAR, which is at 1 meter resolution around a search area. This was about a half a mile. But as you can see here, LiDAR doesn't penetrate concrete. So we still are, if we were to create a streamline network, water would hit that artificial barrier, so the road, and it would back up and then try to find the path of least resistance around the barrier. But we know there's a culvert there, so we have to burn a path for water to pass through underneath the road itself. So to automate this process, we create another search area, which is 100 feet of the culvert, and we calculate the fill, or the depth of each of those cells. So that's just filling up the DEM and then subtracting the original from the filled, and then converting that raster to a point file. So here on this map, you have the road, you've got the culvert as that red circle with the dot in the middle of it, and then you see areas that are deeper with cooler colors and areas that are shallower, so greater than 0 depth, are in red. So we can see there's like water pooling behind the culvert itself. So we use the highway as our center line. That's why that's an input here to define either side of the highway network, and we bisect that culvert buffer area. We select the 2 points of the greatest depth on either side of the culvert, and then just generate a line that goes between it. And this acts as a pseudo barrier, and it burns into the DEM a path that's deeper than the surrounding areas so that water can flow through. So this is the before and after photo. So the before is on the left. We can see that lidar is not penetrating that road, and on the right we have this line now that's burned between there that's going to act as our culvert running underneath the highway system. And then we rerun stream network analysis, which is pretty straightforward. And so now you get this on the left, the original stream network that if you ran and you didn't burn it through, you can see that water again backs up behind the culvert, and it tries to find the path of least resistance over the road. And now we have a stream network that flows correctly underneath the highway. So to evaluate if there's a defined upstream channel, we generate a stream network within that half-mile radius, and if the network is above, has a line network that was above a 2, so a 2 or greater Australia stream order, we say that it is a defined network. The area threshold for this work, I believe, was 1/4 of a mile, a drainage area. So for this here, the red line would be the stream node that we are, the stream reach we're evaluating, that is a 2 Australia stream order of 2 or greater. So we're going to say that yes, it passes that initial flowchart metric. Our next point along the decision tree is to see if there's a high gradient barrier naturally that prevents access to the culvert. So we generate a line segment here that's 100 feet on either side of the barrier, 200 feet on side of either side of the barrier, and then we chunk up the gradient, the lines, and then calculate gradient with those individual segments. Our cutoff here was, I believe, 20%, and so none of these line segments exceed that 20% line, so we say that no, there is not a high gradient barrier that's preventing access, natural access. We also look at searching for this intersection of wetland polygons on both upstream and downstream segments, so it has to pass both tests. It's got to have standing water upstream and downstream. This process did not find upstream downstream intersections, so it intersected with the blue line downstream but not upstream, and so it did not pass those. And all of these decision points, I'll pause here to remind everyone that the decision points provided here were the ones that are actually used in the field by biologists to determine if there are native fish species that are in need of passage on either side of the culvert. And the last thing that we're going to look at is we're using these cutoff values to determine if there are any of one of those 32 species that could access this culvert. And so though we generate our own flow lines that are not present here, we're looking to see if there's any intersection with a natural flow line from the culvert itself. And so in this map, within the half-mile buffer zone of the culvert, which again is that red dot in the middle, we're looking at areas that did have the species, one of these species, which is in green, and those absent species, absent reaches, which are in yellow. And then if the purple line, which is the downstream flow path of this culvert, intersects with a 1 reach, then we would say yes, there is a probability that one of those species could access this culvert. Let's see. And then the last test is looking to see if there's a natural barrier downstream, anywhere downstream to the outlet of this culvert. So we again looked or filtered those barrier data sets by natural barriers that were completely blocked, and we use the trace function in the back side of this tool to look and see if there are any barriers below that culvert. And so here for this test, the culvert is actually that yellow star. Barrier reaches are in red, and the downstream path to that barrier is in green. And so because the green, and I apologize for those of you who are colorblind, but hopefully you can see the different widths at least of the stream lines, is that those two intersect. And so yes, there is a natural barrier downstream of this culvert that would prevent natural movement of native migratory fishes. So as we go through this flow chart again, we would make these decisions. So the tool would loop through each one of the culverts or barriers that you've inputted, and then it gives you an output of yes, this is very likely to have a fish, one of those 32 fishes, and trigger a fish passage law. Probably no, it would not trigger a fish passage law, and unknown is that it could not determine, and it would require a survey check with the district. Now again, this tool is not used to make any sort of final judgments, but rather a scoping tool for crews to go out in the field and say, okay, well, I think this will probably have a fish, which means we might need a little more time to do some significant analysis on it, or probably no, we shouldn't expect that there's no fish here, but still require a in-person check. So the results were really accurate, but in general, when they come out of the output, they look like this, where it has check with the district, there's probably no fish, or yes, it would be a fish call in the final point data set, which all of the attributes that are generated with that final point set show the decision point, again, along this flow chart. So each node on this flow chart has an attribute or a field, and it says, yes, it passed, or no, it did not pass. So if you had concerns and you wanted to know why this tool said, yes, it was a fish call, or no, or where in this flow chart that it landed, you could go back and recreate that. And then lastly, on the back end is there's all those 32 fields with each individual fish species, each of those 32 fishes, and it would give you a probability that that fish was at that culvert or accessing that culvert or not. So you could also check and look at species of concern when you were going to check in that culvert based off the model. So with that, I can take any questions.
Very cool presentation. Seems like it would be a really helpful tool for data processing and kind of streamlining. And you guys can type questions in the chat. You can raise your hand. You can unmute yourself.
I do see a question, let's see, from Denise Kelsey Denise, sorry. They asked if, was the source data to determine if a stream existed above the culvert? So that was the LiDAR data. I will say that, as maybe many of you know, that many of the states are looking forward to replacing the NHD high res with 3D HP. There's really no reason that this tool cannot be transferable to that. It would take a little bit of finagling of making sure that it replaced that trace network with just a regular network that we created, but it can be transferable to different stream networks with a little tweaking, which is the nice thing about having the Python in the background.
Hey, Courtney. I have a question about determining which of these species might be affected. Do you overlay some of this information with the layers of these known fish presences or anything like that?
Yes, a lot of those were taken, the ones that we didn't have enough data for, we took from just like our Oregon fish distribution maps, but those could be subbed in for any fish distribution data set or line network.
Okay.
Hey, Denise, go ahead.
Yeah, hi, great presentation. Sounds like you've been working on this a long time. It's a great product. I do have a question about how you were determining migratory species, because a lot of fish may stay in a single stream, but they're going to go upstream to spawn and come down in the lower reaches to feed. And so I'm just wondering how you decided migratory behavior and whether a culvert is going to affect that species.
That's a great question. I actually did not make that decision. That was based off of the Oregon law. So I'd have to refer you to the legislators to make that map or mind map of how they determined those. But that being said, again, with the tool, you could put in really any species that you wanted to scope in with this tool with like a few little tweaks in the back end.
Okay, great. Thanks.
Courtney, you had mentioned you use the culverts on the highway system. Have you guys thought about looking at other systems like forest roads, like the culverts on forest roads?
No, we have not moved there yet. I don't see why this tool wouldn't work for those as well, as long as, again, you had some sort of forest road line network that you felt was reliable to look on either side of the culvert itself to generate those stream flow lines that aren't currently present on the network itself.
All right. All right. I think we're winding down for questions. Courtney, are you going to stick around in case anybody comes up with questions at the end of today? All right.
Thank
cool.
you.
Thank you. Yeah, thanks. That was a great presentation. All right. So our last speaker today, I'd like to introduce Ryan Vandermeulen. He is with the National Marine Fisheries Service Satellite Coordinator in the Office of Science and Technology at NOAA Fisheries. Before that, he was a senior research scientist with the Science System and Applications Incorporated at NASA's Goddard Space Flight Center. He received a bachelor's degree in marine biology from University of Southern Mississippi and his master's in marine sciences from the same place. Unfortunately, Ryan couldn't be here today for his presentation, but he has provided a recording, which we'll share with you right now. He's also provided his contact information, so if you have any questions, you're more than welcome to reach out to him.
Hi, everyone. Thank you so much for tuning in today. My name is Ryan Vandermeulen. I am the Satellite Coordinator for NOAA's National Marine Fisheries Service. Today, I want to talk to you about a future satellite mission, which will represent a real shift in the way we interact with ocean color data that we use to assess things like water quality, as well as important biophysical interactions in our aquatic ecosystems. I'm very grateful for your understanding in my absence today, but this topic is something I'm quite passionate about, so please do not hesitate to reach out if you'd like to chat more on the topic. I've made sure to put all sorts of fun and colorful animations throughout this presentation to make this seem a little bit less like a pre-recorded talk. Now, for context, just because I'm not necessarily talking to a group of satellite oceanographers today, I just want to give a brief overview of what exactly I mean when I'm talking about ocean color and how it all works. So to begin, the sun puts out electromagnetic radiation that hits the Earth, and all of life on this planet depends on just a tiny portion of the electromagnetic spectrum. Of course, the colors of the rainbow, right? So these are the wavelengths by which plants on Earth photosynthesize, give us oxygen that we breathe. So as this visible light hits the surface of the ocean and goes into the water, only one of two things can happen to that light, as I represent with this friendly little photon here. So the light can either be scattered or it can be absorbed by various constituents in the seawater. Note here that once light enters the water column, if we separate out these various wavelengths of light, we see that light quality changes with depth. Pure water with nothing in it naturally absorbs more red light, which leaves essentially blue light to be scattered back to our eyes. And this is why the open ocean appears blue to us. Some of these photons are lucky enough to be kind of redirected out of the sea surface, back out into the atmosphere and into the field of view of a satellite. So a satellite sensor in orbit is simply measuring the intensity of various wavelengths of light being reflected back. And thus, ocean color science is really just the sort of mathematical, scientific representation of what your eye is already telling you. And here's an example of a satellite snapshot with a wide variation in ocean color over relatively short distance. This is off of the Mississippi River Delta. So we have a lot of materials coming out of the river that we can see floating in the water, as well as micronutrients that are fueling the growth of phytoplankton in the ocean. So you can see here, of course, that the water appears kind of brown near shore, and if we move offshore, it becomes more blue. Now, if you look at the intensity of light, in other words, essentially what a satellite sees, it might look something like these panels on the right. These represent the intensity of light from blue on the left to red on the right, and you can see that the shape of this intensity changes over the spectrum. So in green waters, the peak of intensity is over the green portion of the spectrum, and so forth. So satellites are looking at the entire surface of the ocean and giving us millions of unique optical fingerprints, or these distinct combinations of colors that are being reflected from the ocean surface. Now, in order to make this information useful, we have to decipher what specific components of the ocean water are causing the color to change. And it could be any combination of microscopic phytoplankton, it could be sediment or dead cells, floating seaweed, microplastics, pollution, dissolved river runoff, oil slicks. All of these things have their own unique optical fingerprint based on their presence, their size, shape, abundance, and overall composition. So if we can, from a ship, establish a clear relationship between the different components of the ocean and assign it a specific optical fingerprint, we can essentially look for this signal anywhere on the globe and then infer that the contents of the water are similar. How well we can separate out this information from satellites is largely reliant on how close we can zoom in to see the materials, the range and specificity of colors that we are detecting, as well as the sensitivity of the satellite instrument. Superimposed on this globe is a map of the photosynthetic pigment chlorophyll A changing over time and space. Now on land, we know chlorophyll is this pigment that keeps grass green, but on the ocean, it can be found in dense concentrations, and it represents the machinery that fuels the growth of phytoplankton, forming the basis of our entire marine food web. The satellite remote sensing of ocean color has really been revolutionary in providing an unmatched insight into information about algal biomass as well as biophysical interactions. And this is really our only global insight into trophic energy as well as carbon export dynamics that we can observe at this scale. So this synoptic vantage point really lends tremendous value to helping understand hotspots where marine species live, eat, and migrate, as well as making predictions about future productivity of fisheries in response to environmental change. There's a lot more to the color of the ocean than meets the eye. As we mentioned before, other things can change the color of the water. For instance, harmful algal blooms such as red tides are becoming more ubiquitous, and a single event can cause billions of dollars of damage to localized economies, from losses to tourism and fisheries to cleanup costs as well as human health impacts. Now, over longer time frames, we're even seeing that this has an impact on the real estate market, as well as people's willingness to, say, book a vacation at a particular location. So state and local agencies often don't have the resources to be able to, sample for various parameters that impact their water quality, or at least not very often. But by using satellite ocean color information, we may be able to save hundreds of millions of dollars in environmental monitoring costs alone. And there's also means where we could help potentially use this information to help fishermen avoid areas where we're most likely to encounter a certain species that we want to avoid. So it really begs the question, why don't we have more ubiquitous weather forecast analogs for the ocean? Why is our predictive capability seemingly lagging relative to other scientific disciplines. Now, to help answer this, I want to talk about some of the technical details of the choice of a satellite orbit. Now, colloquially, when we're talking about ocean color, we're referring to measurements that are collected by polar orbiting satellites, which, as you can see here, means it is able to circle around the globe along a vertical axis, measuring from pole to pole, and eventually giving us a global snapshot of the Earth. Now, how much of Earth is covered in a single day is largely a function of how far we want to zoom in. In general, the more detail you want equates to longer periods of time to get a global picture. Alternatively, we have what is known as a geostationary orbit. This is the type of orbit that our communications and weather satellites fly at. They're located about 50 times further from Earth than the polar orbiting sensors, so they collect data a little differently. Here, we're orbiting along a horizontal axis along the equator, but the orbit speed is the same speed as the spin of the Earth. So from the satellite's perspective, Earth is essentially standing still. What this allows us to do is get multiple views of the Earth on a given day. And this is how we can create things like, movie loops of hurricanes and storms. Since 1975, NOAA has been operating a series of geostationary sensors that largely have been dedicated to serving the weather mission. Now that's changing. In the coming decade, this mission will evolve into a full environmental monitoring suite, examining air quality, water quality, fires, lightning, floods, volcanoes, and in general, just offer information at a quality and cadence necessary to enhance our numerical prediction models for all of these aforementioned parameters. So together, these various instruments, work to address all of NOAA's key mission areas, climate to a weather-ready nation, resilient coasts, and healthy oceans. Now, included in this suite is an instrument for measuring ocean color. And this would represent a new capability for the United States in providing this type of data at a higher resolution and cadence than we have yet to accomplish. So just to give you an idea of how this would work, there would be two ocean color instruments in orbit at the same time. One looking at the East Coast, Great Lakes, Gulf of Mexico, Puerto Rico, and the other looking at the West Coast targets, Alaska and Pacific Islands. This represents coverage of the entire U.S. exclusive economic zone. Now, we'll get into this more, but this instrument will have spectral characteristics such that we can really hone in on that optical fingerprint I mentioned before, and then we'll also be able to do so at scales that are intended to be more relevant to coastal communities. So this is not going to be a sensor strictly for global carbon monitoring. We're going to leave that to our capable fleet of our polar orbiting sensors. This is the type of sensor that after a hurricane passes through or an oil spill happens, we can find areas where currents are converging and likely accumulating marine debris. This is also a sensor that can enable point source detection of hazardous pollutants and allow us to track the optical fingerprint of freshwater inundation after a weather frontal boundary moves through. This is also something that conceivably can help shellfish farmers make decisions about harvesting if there's a proximal harmful algal bloom in the region. Now, these sort of applications are not an afterthought of the mission. Everything about the ocean color instrument specification and its requirements are specifically designed with these sort of applications in mind. Applications concerning public health, food safety, recreational activities, hazard mitigation, environmental impacts, ecosystem health. Some some novel capabilities will become possible by just having the availability of these sequential images. So you can make movies. This can help us, for instance, track surface currents and other kinetic processes. But more generally, what we're gaining from a geostationary perspective is really better coverage and consistency in our measurements, that the kind of measurements that live up to the standard for data assimilation into our predictive models. All right, so what is it about this geostationary orbit that changes this so drastically? All right, let's take this one enhancement at a time here. And we know the Earth is a cloudy planet, and from the perspective of space, clouds are completely opaque. This presents a big problem for satellite oceanographers because we can't see the ocean underneath. Current satellites are passing over the Earth once per day, and the majority of data over the ocean is basically discarded because the data is contaminated by clouds. The image on the top left just gives you an example of this daily snapshot that we get, in this instance coming from SNPP VIIRS mission. By contrast, if you look at the lower panel, an instrument like GeoXO will obtain observations every 2 hours, and as the clouds pass by, we can fill in those gaps to double or even triple the amount of ocean that we see every day. This image on the bottom right was taken from a Japanese geostationary instrument, and what you see is a composite of the same exact day as the image above. And you can clearly see that a more cohesive picture of the ocean emerges. More data helps feed our models with gap-filled, up-to-date information, which is really critical if you're using data for decision making. You need to know the data stream is reliable and consistent. Now, our current constellation of ocean color satellites collect data by essentially sweeping the sensor left to right, and so it can be more efficient in covering more area. Now, the image on the left is an example of what our typical 1 kilometer spatial resolution looks like when the satellite is looking straight down at the Earth. Now, 1 day later, the same sensor passes over, but the satellite was in a different position, and it collected data on the high end of one of these sweeps. So it's looking at the Earth at kind of an angle. In the middle panel, you can see the impact on data quality as the size of the pixels grow by about a factor of 4. Now, in a geostationary orbit, we are just that, stationary. So the pixel size at any given location will never change over the lifetime of a mission. It is forever consistent. What's more, since we're in a geostationary orbit, we have the chance to dwell on the Earth and collect more signal, which means we can zoom in closer and get more details of these coastal processes without our data becoming too noisy. The image on the right shows an example of the approximate spatial resolution, or the detail that we can anticipate from this GeoXO mission, providing details of the really the aquatic environment that are more relevant to these nearshore processes. So moving on, for the last 27 years, the colors that satellites look at are discretized segments of the visible spectrum, essentially taking 5 to 10 subsamples of the colors of the rainbow. You might be thinking, cool, there's only really 7 colors of the rainbow anyhow, but there's a lot of unique information in between those gaps of color. If we refer back to the optical fingerprint analogy, if you get a partial print, you only get a partial answer. And the more features you can identify within that print not only ensures a higher degree of confidence, but also may contain information that helps distinguish two things that may look similar but aren't. The Ocean Color instrument on GeoXO is a specialized hyperspectral instrument, meaning that it can measure optical details of the ocean very precisely. Capturing these fine details will enable new insights into phytoplankton community composition, which forms the basis of our entire marine food web. different phytoplankton have different nutritional values, and they introduce different amounts of energy into the aquatic system. This might not sound like too big of a deal, but climate-related shifts in phytoplankton communities have caused an entire menhaden fishery to crash in the northern Arabian Sea some seasons. So this is really important to keep track of. And knowing more information about the composition can also help us determine if a particular phytoplankton bloom is likely to be toxic, which can cause mass die-offs of wildlife in the ocean. Now, sometimes it's not only about being able to sense them with color, but logistically also being able to see events often enough to know when conditions are different. Now, we're in the process of publishing results from a study showing that when we hold all other things equal, because geostationary measurements allow us to get more frequent, high-resolution measurements, we're able to detect hundreds of more anomalous water quality events over the course of a decade. So if you're monitoring a sensitive ecosystem like a coral reef, there's not much sense in relying on satellites if we can't see events in a time frame that allow us to make decisions or take action. That's really the bottom line here. This is just a little movie I like to show that illustrates the enhanced detail we'd be able to see when our focus is centered on near-shore observations and coastal communities. It's this level of detail here is representing ocean processes that are relevant to the distance that a small fishing boat can travel over the course of an afternoon. The simple act of being able to conceptualize information better really provides immense value to our potential stakeholders, because we could start asking and answering questions that are relevant to scales that impact us more acutely. So now we're in the process of mission advocacy and building up that stakeholder community. Now, not all the ink has dried on the details of this mission, so it's important that we are really connected with our users at this stage so that they can provide critical feedback on design choices and so forth as we move forward. From fish finding services to defense agencies, water managers, ecosystem modelers, and even insurance agents. We've got a group of folks who are really hungry for this type of data stream. Now, this particular mission doesn't launch until 2032, but in the context of a satellite mission timeline, really between now and the next year is the time when our user community input is really critical. So as I've been kind of going off about all the advantages of this particular GeoXO mission, in the end, the majority of our users really don't care where the data comes from or who serves it up, just that the products are readily available. To that end, it's not just one single satellite mission really solving all of the world's problems. There's this whole constellation of emerging satellite capabilities coming within the next decade. Last year, NASA launched the PACE mission, which is our first global hyperspectral ocean color mission. Another NASA mission, Glimmer, will be launching in 2028. This is actually going to have pretty similar capabilities to GeoXO and allow us to really get our hands dirty with this type of data, but it's going to have a reduced lifespan relative to the 20-year lifespan of GeoXO. Now, other missions like SBG and Landsat Next are also underway, and they'll be giving us hyperspectral imagery at even higher resolutions to where we can classify benthic habitats and differentiate types of seagrass. It's really a global synthetic community of satellites supported by multiple space agencies. And the science that satellites are providing is also supported by a vast network of in situ platforms and on-the-ground sampling, which are really important to help us verify and learn more about life on our blue planet. I'm just going to leave you here with a new movie, a promotional movie about the GeoXO mission.
Is orbiting Earth to keep constant watch over the Western Hemisphere. This new generation is made up of 3 satellites: GeoEast, GeoCenter, and GeoWest. The 3-satellite constellation is measuring, monitoring, and watching events across the United States, all at the same time. Using data from current satellites, we can imagine what this new generation will see. On this particular day, the constellation is using the atmospheric composition and the infrared sounder instruments to monitor air quality. Today, the air quality is particularly poor in the Los Angeles metropolitan area. Health officials use the satellite data to issue an air quality warning, encouraging residents to remain indoors or limit their time outside. The satellites are also informing communities and first responders further north, where thunderstorms are rolling through. The GeoXO sounder is monitoring the region and alerts forecasters to the potential for severe weather. The GeoXO imager is providing near real-time, high-resolution imagery of the storm, and the lightning mapper detects an increase in lightning activity as the storms strengthen. As day turns into night, the storms become stronger with an uptick in lightning in areas packed to the brim with dry vegetation. Concern for the development of wildfires grows. With the help of GeoXO, the National Weather Service and local forecasters warn the public of the storms and alert firefighters to the potential ignition of wildfires. Multiple fires have ignited. The GeoXO imager monitors the size, intensity, and movement of each fire, helping to keep wildland firefighters and nearby communities safe. The smoke produced by these fires is spreading across the U.S., causing the air quality to worsen. The atmospheric composition and imager instruments help to inform vulnerable communities about the thick smoke and poor air quality. While GeoXO is helping fight fires and smoke in the west, another threat is forming in the Gulf of Mexico, just offshore of Alabama, Mississippi, and Louisiana. Recently, runoff from flooding due to a hurricane stirred up bacteria in the water. The area is also affected by chemical runoff from farther upstream. Using special filters within the GeoXO ocean color sensor, scientists determine what species of algae is blooming and produce models to measure the extent of the bloom. Today, the ocean color sensor detected the potential for the presence of cyanobacteria, the toxic kind of algae. Local officials use this data to issue public beach warnings to prevent people from getting sick. This early warning can also help drinking water utilities take evasive action to keep tap water safe from toxins. After one storm stirred up sediments off the Gulf Coast, another is turning in waters farther south. In the Gulf of Mexico, GeoXO satellites monitor a hurricane headed for southwest Florida. Together, the imager and lightning mapper provide minute-by-minute monitoring of the structure of the storm, lightning activity within the storm, and path the hurricane takes. The sounder further improves the hurricane track and intensity forecast. Sounder data gives insights into the winds and the clear air surrounding the storm, not just near the tops of the clouds. This data helps models and forecasters to predict the future movement of the storm. Along with forecast model information, this prompts officials to issue an evacuation order to residents likely to be affected by the storm. The GeoXO instruments work together to provide near real-time information to forecasters, decision makers, and first responders. From severe weather and hurricane warnings to air quality, wildfires, and marine hazards, GeoXO contributes to the protection of life and property within the United States, all in a day's work.
So thank you all so much for listening. My email is up top here. Please don't hesitate to reach out if you'd like to chat more. Thanks.
All right. I have dropped Ryan's email in the chat too, if that's more helpful to copy paste that into your email addresses. All right, and like I mentioned, Ryan wasn't available to join us today, so send all your questions his way. Otherwise, we'll go ahead and open up the floor again for questions on either Ed's presentation or Courtney's. All right. Sam, seems pretty quiet. You want to do wrap up?
Yeah. Yeah, thanks everyone for joining us today, and a big thank you to Ed, Courtney, and Ryan. Great presentations, and as Meg mentioned, I will follow up with an email, which will include a link to this recording that you can share amongst your colleagues. And so, yeah, this was the last week of our webinar series. So nothing to look forward to next week, except keep an eye out for a survey that will be sent out to everyone who joined these ETIS webinars. We will be doing more of these in the future, and we need your input to kind of form how we're going to do this next time. And a big thank you to this ETIS planning committee that I have here. Like I mentioned at the beginning of this presentation or of this session, we didn't start from scratch. It took a lot of people work from years past to get this going. So thank you to everyone of them. And yeah, that is it. I will stick around for a little bit longer if anyone has questions about the ETIS webinar series. But thank you for joining.
Thanks, everybody.